Good morning, everyone. We are starting now the webinar. I'm very happy to host this webinar by David Vilchez. The seminar by David Vilchez was scheduled for March, but due to the COVID-19 outbreak, we had to cancel it, and now we are hosting it as a webinar. I would like to introduce David. He and a bit his professional timeline. He did his PhD at University of Barcelona in Spain. After that, and a short postdoc stay right there at the Institute for Research in Biomedicine in Barcelona. He moved to the Salk Institute at the United States as postdoc, and to University of California as a research associate and associate specialist until 2014, when he moved to the University of Cologne, Germany, where he is currently a group leader and principal investigator. He works at the Institute for Genetics and Cluster of Excellence, Cellular Stress Responses in Aging-Associated Diseases. He has a very brilliant track record. He works, as we will learn today, he works on proteostasis, and he has conducted groundbreaking research in this field in relation to aging, neurodegeneration, and stem cell biology. He has publications in top-ranking journals: Nature, Nature Metabolism, Nature Neuroscience, Nature Communications, and other high-impact journals. He currently holds an ERC starting grant, and he has also attracted important external funding from the German Research Council, Marie Curie Career Integration Grant, and other grants. So today he will give the seminar on proteostasis of aging and stem cells, and I hope we don't have any technical problems with this webinar. You will be able to ask questions via the chat in YouTube during the seminar, or at the end. After his seminar finishes, I will read the questions from the chat to him. And thank you. I will now give the floor to David for him to start this his presentation. David, when you are ready.
Creo que sí. Bueno, ¿se me escucha?
Okay, I will now close my camera and I will silence the microphone, and you should be able to share your screen if you have any problem. We hope we'll be able to solve it.
Okay. So, thank you so much, Lourdes, for the introduction. Parecía que hubo algunos problemas con la conexión, pero creo que ya se han solucionado. Everyone can see my screen? I hope so.
Yes, yes. We can see it, and I will check now at YouTube, but I think there's...
Okay, so I will give the talk in English, not because I like it, it's because, you know, after that many years outside or abroad, it becomes very difficult to give a seminar in Spanish. So first of all, I would like to thank Lourdes and the CBM for this opportunity to present our work. And as Lourdes said, I was supposed to visit the CBM in Madrid, and actually I was really looking forward to going to Madrid, enjoying the good food and visiting you in person, but of course that wasn't possible. But anyways, I'm very happy to have this chance to present our work online. So today I'm going to talk about our approach to study stem cell identity and the aging process. So let me see if I can put this fancy on. About our approach to study stem cell identity and the aging process. So our project is based on the basic observation that protein homeostasis or protostasis declines during the aging process. So this decline in protostasis can promote the accumulation of damaged, misfolded, or aggregated proteins, affecting cell function, and eventually leading to disorders such as Huntington, Parkinson, or Alzheimer's disease. So the key question of our laboratory is if we can postpone these diseases and slow down the aging process just by modulating protostasis. And to address this challenge, we propose something completely different. We propose to use human embryonic stem cells because we believe that these cells could provide a unique system to study protostasis. First of all, they can replicate forever in vitro without any sign of aging, so they never age. So they are considered to be immortal in vitro. And second, they can differentiate into any cell type of the organism. So we believe that stem cells must have an increased protostasis to maintain the cell renewal, the immortality, and also to generate new cells with an intact proteome, because otherwise that could affect development and even the aging process. So basically what we do is to define how stem cells regulate the protein homeostasis, the protostasis, and how this protostasis regulates stem cell function, differentiation, and immortality. So then the idea is to apply these findings into the somatic tissues like neurons, muscle, etc. of model organisms, mostly in C. elegans, to try to extend longevity and delay the onset of age-related diseases. So at the very beginning, actually during my postdoc, we focus on only one aspect of the protostasis network. We focus on the proteasome because this node is critical for maintaining the proper concentration of many regulatory proteins and also to terminate damaged proteins that are prone to aggregation, so they must be scavenged. So our project started with a very basic question. We asked if stem cells have an increased proteasome activity compared to their differentiated counterparts. In other words, if they lose the high proteasome activity when they differentiate. So what we did is to have several stem cell lines. We differentiated them into neural progenitors that are still multipotent. They can differentiate into astrocytes, neurons, oligodendrocytes, and they still can proliferate. And then we finally terminally differentiated these cells into neurons. So as you can see here, when we differentiate stem cells into neural progenitors, there is a massive decrease in the proteasome activity. That was further decreased when the progenitors differentiated into neurons. So it seems that the more differentiated, the less proteasome activity. So here I'm showing neurons, but I should say that that also happens when we differentiated stem cells into any cell type, like muscle, fibroblasts, trophoblasts, etc. So then we ask, okay, can proteasome activity be reprogrammed? So for that, as you know, so from somatic cells, I can hear something in the background, sorry about that. So from somatic cells such as fibroblasts, we can generate induced pluripotent stem cells that are very similar in many aspects to embryonic stem cells, like in the ability to differentiate, proliferate, in the transcriptome, etc. So now we wanted to know if they also have an increased proteasome activity. So what we did is to reprogram fibroblasts into two independent iPS cell lines. And as you can see here, both iPS cell lines have much more proteasome activity compared to the parental fibroblasts. And actually, this proteasome activity is very similar to the one that we can find in human embryonic stem cells. So we can conclude that indeed proteasome activity can be reprogrammed. So then we ask, okay, is this high proteasome activity required for the potential supervigilant protostasis of stem cells? So here we focus on Huntington disease. That is a disorder caused by mutations in the Huntington gene that spans the polyglutamine repeats in the Huntington protein. So basically, more than 35 polyQ repeats will promote the aggregation and toxicity of the protein. But what is important to remark here is that Huntington disease or iPS cells from Huntington disease lines behave completely normal. They behave as control cells. So they can proliferate, they can differentiate into neurons, but once differentiated into neurons, they start showing disease-related changes. Like, for instance, they become more sensitive to cellular stressors. So Huntington disease is an autosomic dominant disorder. So our iPS cells have one normal copy of the Huntington gene and one mutant copy. So by using an antibody that can discriminate between the two forms, we confirm that indeed Huntington disease iPS cell lines express significant amounts of mutant Huntington, but even with these significant amounts, we couldn't find a single cell with toxic aggregates. So maybe here is more clear. We have two independent Huntington disease lines expressing different mutant alleles. And as you can see, there is no cell with aggregates. So they are like controls. None of them accumulate aggregates. But when we treated the cells with proteasome inhibitors, then the Huntington disease lines, but not the control lines, then this Huntington disease line started to accumulate aggregates. And later, we found that this aggregation phenotype correlates with an increase in the total levels of mutant Huntington. So we can conclude that this high proteasome activity of stem cells indeed prevents the aggregation of mutant Huntington in these cells. So the question now is how stem cells achieve the high levels of proteasome activity? Well, active proteasomes, also called 26 or 30S proteasome, are formed by the interaction between the 20S proteasome that contains the catalytic activity with the 19S regulatory subunit that is responsible for stimulating the 20S to degrade proteins. So it's considered that these free 20S are inactive in cells because their channels are closed, and they need, for instance, to bind to the 19S in order to open their channels and be able to degrade proteins. Sorry. So then by native gels, we assess the assembly of the proteasome, and we found that when stem cells differentiate into neurons, there's an increase in the levels of free 20S, which is the inactive form of the proteasome. In contrast, the most active forms of the proteasome, especially the 30S, decrease during differentiation. So with these results, we thought that that could be due to maybe an increased assembly factor, or maybe they just have more 19S subunits. And actually, we found that it's a combination of both, because when stem cells differentiate into neurons and we analyze the levels of all the proteasome subunits, we found that only one of them decreases during differentiation. And this one is RPN6. So as you can see, it decreases at both the transcript and protein levels. And these results were very striking because RPN6 was the only proteasome subunit that decreases during differentiation into any cell type, like for instance, into fibroblasts or trophoblasts. So why RPN6 could be that important for the assembly of the proteasome? So first of all, RPN6 is part of the 19S lead. Actually, it's essential for the assembly of the lead. But the most interesting for us is that RPN6 interacts with an alpha subunit of the 20S and with a subunit of the 19S base. So RPN6 is like a scaffolding protein that promotes the interaction between the 19S and the 20S, and then forming more active proteasomes. Indeed, we found that when we overexpress RPN6 in 293 cells that have relatively less proteasome activity compared to stem cells, we found that this overexpression was sufficient to increase proteasome activity in these cells, and that correlated with an increase in the assembly of active proteasomes. On the other hand, if we knock down RPN6, the cell loses the proteasome activity, and that was associated to a decrease in the assembly of active proteasomes. So we can conclude that RPN6 is both sufficient and necessary for increased proteasome activity. So then we ask what could be the role of RPN6 in the context of aging, and for that we use C. elegans as a model organism. And what we did is to overexpress RPN6 only in somatic tissues, only, for instance, neurons, muscle, etc. And we found that by overexpressing RPN6, we could increase proteasome activity, and that resulted in a lifespan extension of up to 25%. That is quite significant. But the most striking is that RPN6 overexpressing worms survive much longer under stress conditions, like heat stress or oxidative stress that put stress on the proteins. So when we overexpress RPN6, you can see that they survive much longer compared to the control strain. So with the result, we asked, okay, is RPN6 sufficient to protect or prevent toxic protein aggregation? So for that, we use a Huntington disease C. elegans model that expresses polyglutamine repeats only in the neurons. So when the worms accumulate these toxic aggregates, they lose the motility. Actually, they become almost paralyzed because these aggregates induce neurotoxicity. But we found that just by overexpressing RPN6, we could rescue this motility phenotype. And later, by filtered trap assays, we found that RPN6 overexpression decreased the accumulation of aggregates in neurons. So to summarize this first part, we have seen that by using stem cells as a model, we define RPN6 as a key activator of proteasome activity and assembly, that then it can be mimicked in somatic tissues to extend longevity and delay protein aggregation. So that was very important because actually RPN6 was the first activator or known activator of the proteasome. But here, at the same time, there's a problem because, as you know, the proteasome not only regulates damaged proteins, it can affect the concentration of many regulatory proteins. So if we are just activating proteasome activity globally in the cell, it could be that this activity is also affecting other pathways, like for instance, apoptosis, cell cycle, or proliferation. So now we wanted to define a more specific mechanism that can prevent the aggregation of damaged proteins without affecting other pathways. And for that, we focus on E3 ubiquitin ligases. Why? Because these E3 ligases transfer ubiquitin to a specific target, and then this target protein can be recognized and degraded by the proteasome. So by combining proteomics and transcriptomics, we identify 26 E3 enzymes highly expressed in stem cells compared to neurons, most of them of unknown function. And after we validated them by Western blot, we asked if they can prevent from polyQ aggregation in Huntington disease iPS cells. So we already obtained interesting results with one of them, with UBR5. So we found that indeed UBR5 decreases in a progressive manner during differentiation into neurons. But the most important is that UBR5 interacts with both with normal Huntington and also with mutant Huntington. And then when we knock down UBR5 in Huntington disease iPS cell lines, then we observe a huge increase in the levels of mutant Huntington. And of course, that could be relevant for aggregation. Indeed, when we knock down UBR5 in Huntington disease lines, then these cells started to accumulate aggregates, as we confirmed by immunostaining and also by filtered trap. In contrast, the control lines didn't accumulate aggregates because they don't express the mutant Huntington. So then we ask, okay, how relevant is UBR5 for neurons, which are the cells that are more affected in Huntington disease? So what we did is to use again our C. elegans model of Huntington disease. We knocked down UBR5 and we found that this induces a dramatic increase in the aggregation of polyQ peptides. And that translated into a further decrease in the motility phenotype because we are increasing the toxicity induced by polyQ aggregation. So then the next logical question was if UBR5 can be sufficient to prevent mutant Huntington aggregation. So here we had a technical problem because UBR5 is a huge protein of over 300 kilodaltons, so it was impossible for us to overexpress it in C. elegans. Believe me, we tried many times. So for this reason, we decided to generate human cell lines. And as you can see, when we overexpress mutant Huntington with more than 100 polyQ repeats, then we got a very strong aggregation phenotype. if we express normal Huntington, then we don't see aggregation. But the most interesting is that when we overexpress UBR5 at the same time, wild type UBR5, then we could decrease the levels of mutant Huntington, and that, of course, ameliorated or decreased the aggregation phenotype. But when instead of wild type UBR5, we overexpress a catalytic inactive mutant form of UBR5 that doesn't have E3 ligase activity, then we couldn't decrease the levels of mutant Huntington, and also we couldn't ameliorate or improve the aggregation phenotype. So these results with the mutant are very important because that indicates that UBR5 is actually inducing or directly inducing the polybiquitination of mutant Huntington. So to test that, what we did is to perform immunoprecipitation experiments of Huntington. But first of all, we treated the cells with proteasome inhibitor to avoid any degradation of Huntington. So as you can see, the levels of immunoprecipitated Huntington were very similar in all the conditions, but only when we overexpressed UBR5, we were able to see a huge increase in polybiquitination. In contrast, if we overexpress the mutant UBR5, we didn't see this polybiquitination. So finally, we confirmed that this polybiquitination induced by UBR5 promotes the degradation of mutant Huntington through the proteasome. So for instance, here you can see that when we overexpress UBR5, we decrease up to 50% the levels of mutant Huntington. But if we treated the cells with proteasome inhibitor, we completely block this aggregation, this degradation of mutant Huntington. So indeed, it seems that UBR5 modulates mutant Huntington levels through the proteasome activity. So to summarize this part, we have seen that iPS cells have or express high levels of UBR5 that polybiquitinate mutant Huntington, and then mutant Huntington can be recognized and degraded by the high proteasome activity of stem cells. So that results in decreased levels of mutant Huntington that then can be easily controlled by other proteostasis mechanisms that then completely prevent the aggregation of mutant Huntington. So this is very interesting, but I would like to note that iPS cells, even with these high levels of UBR5, they still express significant amounts of mutant Huntington. So now we wanted to define which are these other proteostasis mechanisms that specifically prevent the aggregation of the polyQ expanded Huntington. So for this, we perform a comprehensive analysis of the entire proteostasis network in stem cells, and we already identify many proteostasis components in stem cells that are increasing stem cells compared to differentiated cells, and for instance, components of the unfolded protein response that assure the proper folding or degradation of damaged proteins. But what really drew our attention was that several subunits of the trig complex decrease during differentiation. So the trig complex is a chaperonin that regulates the folding of proteins, and it contains two stacked rings formed by 8 subunits each, with a cavity inside for the folding of newly synthesized proteins. So it's considered that the trig complex is involved in the folding of around 15% of the proteome, which is quite a high percentage, and it's also known that it can regulate protein aggregation in Huntington and Alzheimer's disease models. So you can imagine that with this important role of the trig complex, loss of CCT subunits can decrease cell proliferation and viability. And another important characteristic is that CCT subunits, the expression of CCT subunits, decrease during the aging process in somatic tissues, for instance, in the human brain. So for all the reasons, we thought that it could be very interesting to study the trig complex in the context of pluripotency. So, as I said, different CCT subunits decrease during differentiation into neurons, like CCT8, CCT7, of CCT2, and others remain similar. So here, again, I'm showing neurons, but that happens when we differentiated them into any cell type, I mean, mesoderm, endoderm, etc. So then what we did is to induce a single knockdown of the different CCT subunits. I mean, each by separate in the Huntington disease iPS cell line. So as you can see, knockdown of any of these CCT subunits was sufficient to trigger aggregation in these Huntington disease iPS cell lines that normally, in normal conditions, they don't accumulate aggregates. But in contrast to what we observe with proteasome inhibitor or UBR5 knockdown, in this case, loss of CCT subunits doesn't affect the total levels of mutant huntingtin. So that indicates that CCTs or the trig complex is specifically regulating the aggregation step. Another important conclusion of these experiments is that loss of different CCT subunits eventually induce polyQ aggregation. So since it's known that loss of a single subunit decreases the assembly of the trig complex, we thought that these effects are actually modulated by an increased assembly of the complex rather than any monomeric function or potential monomeric function of CCT subunits. Indeed, by native gels, we found that the stem cells have an increased assembly of the trig complex in the form of 2 stack rings, and they lose this high assembly when they differentiate. So the main question now is how stem cells activate the assembly of the trig complex. So here we went back to our favorite model, to see Elegans, and first of all, we confirmed that somatic tissues express less amounts of CCT subunits compared to the germline, like that happens, for instance, in the intestine, neurons, etc. So that indicates that this C. elegans could be a good model for our project. So what we did is to overexpress only in somatic tissues those CCT subunits that are normally increased in human embryonic stem cells. So as you can see, we could obtain overexpression levels of all of them, but only overexpression of CCT8, here this red line compared to the black line, only CCT8 was sufficient to induce a lifespan extension at 20 degrees. That is the normal standard condition or temperature in the lab. But these results were even more spectacular when we put the worms at 25 degrees. That is a mild, moderate heat stress condition. So at under 25 degrees, the worms live up to 40% longer compared to the control strain. So then we could completely block this lifespan extension when we knock down a different CCT subunit, like CCT6. So you can see that if we knock down CCT6 in the CCT8 overexpressing worms, we could decrease the longevity phenotype, but if we knock down CCT6 in the control strain, we didn't see any effects on lifespan. So that indicates that CCT8 is promoting or inducing longevity through an increased assembly of the trig complex. As we confirmed by native gel analysis, so here you can see that when we overexpress CCT8 in the somatic tissues of C. elegans, we could increase the assembly of the complex in the form of 2 stack rings. So then we asked if these effects are evolutionarily conserved. And for that, we overexpress CCT8 in human cell lines. And as you can see, we obtain a very strong overexpression levels. But the levels of other CCT subunits, the total levels of other CCT subunits didn't change. But by native gel, we found that just by overexpressing CCT8, we could increase the assembly of the trig complex. And in contrast, the levels of other CCT subunits, the free monomeric levels of other CCT subunits, decrease because priori they become part of the trig complex. So with the result, we thought that CCT8 could be an interesting candidate to be modulated in the context of aging. So as I said, it's known that CCT subunits, the expression of CCT subunits decrease with age in the human brain. And actually, we found that this also happens in C. elegans. At day 5, there was already a significant decrease in the expression of CCT subunits. So we thought that this decline in CCT subunits could contribute to loss of protostasis. So we thought, okay, so maybe we overexpress CCT8, we can reverse or block this decline in protostasis. So what we did is to induce heat stress, prototoxic stress at different ages. So when we induce heat stress at day 1, we found that CCT8 overexpressing worms don't show any effect. They live quite similar, or they survive quite similar compared to the control strain. But if we induce heat stress at day 5, when the worms already lose the protostasis, then CCT8 overexpressing worms survive much longer compared to the control strain. So that indicates that CCT8 overexpression can be sufficient to sustain the loss of protostasis during the aging process. So finally, we found that when we overexpress CCT8 in the Huntington disease model, we could decrease the accumulation of toxic aggregates, and the worms also become much more happier. They start moving again because we are decreasing the neurotoxic effects induced by polyQ aggregation. So for this part, I would like to thank these very talented PhD students that were such a good friends that they even match clothes, so don't even ask. But I hope this, I know this picture is disturbing, but still I hope that I could convince you that stem cells are a good model to study protostasis, because by using these cells, we have defined RPN6 as a key activator of proteasome assembly, UBR5 as a modulator or stimulator of degradation of damaged proteins, and CCT8 as a central activator of the assembly of the trig complex. So then, if we mimic these protostasis components in somatic tissues of model organisms, we could extend longevity and delay the onset of age-related diseases. So this is, I know it was maybe too much information, but this is a major area of our laboratory. I hope you found it interesting. But now I would like to completely switch gears and talk about a different project that it was, I think, is also very exciting. And I must say that this project started more for fun when I was bored of writing grants, etc. So I was doing my little experiments, and then it became to be very interesting. So I'm very happy to share this project with you, or these results. So in this other project, we wanted to study how the reproductive system regulates the aging of the entire organism. So again, this project is based on the basic observation that with age, there is a decline in somatic function that can lead to disability, the onset of age-related diseases, and also to an increase in the risk of death. But at the same time, there's also a clear decline in fecundity that, of course, contributes to the demise or decline of reproductive capacity during the aging process. So our goal was to define mechanisms that can prolong both longevity and fecundity. So if you follow the current paradigm, that will tell you that this is not possible, because in many cases, fecundity and lifespan are negatively correlated. In a range of organisms, from invertebrates to mammals, long lifespans are usually associated with low fecundity, and actually interventions that decrease fecundity can extend lifespan. And this could be explained because in nature, organisms have a limited amount of metabolic resources or energetic resources, and they have to invest these resources to maintain the integrity of tissues that are constantly challenged by the environment, by metabolic conditions, or by pathogens or in disease. So, of course, organisms or animals have somatic tissues and also a germline required for reproduction. So with this limited amount of resources, the organism have to decide between distributing them for somatic maintenance or for germline fitness and reproduction. So the disposable soma theory of aging will tell you that organisms normally follow a trade-off strategy because they invest a higher percentage of the metabolic resources to protect and prevent damage to the germline, so they can assure that they can reproduce and have a healthy progeny. On the other hand, the somatic tissues receive less energy or nutrients and metabolic resources, so they deteriorate, and that contributes to the aging of the organism. So this has been extensively studied in many modern organisms. For instance, in C. elegans, mutations in the GLUT1 gene completely remove the germline in adults. So these mutants are basically worms with no germline, and they live up to 60% longer compared to the control strain at 20 degrees. So this led to the hypothesis that the germline can actively promote somatic aging by releasing pro-aging signals or signals that stimulate the aging process, or by inhibiting pro-longevity mechanisms. So we have done important contributions to this field because we have seen that in GLUT1 mutants, the ones that don't have a germline, the somatic tissues have much more proteasome activity compared to the control strain, and also they have increased expression of chaperones that eventually or more directly contribute to the longevity phenotype. So this is a very nice hypothesis or important hypothesis to explain why we age. But here we challenge this view by asking what happened under favorable conditions. So in other words, why the organism should sacrifice either the soma or the germline when these advantageous conditions probably will allow to keep both. So the question now was, okay, what could be these advantageous conditions where we can study or assess our hypothesis? So I must say that the idea came in a peculiar way. So don't get me wrong, I was born and raised in Barcelona, then I did my postdoc in San Diego, and then I moved to Cologne. So I love the city, it's a very nice city, people are very nice, but let's say that the weather is not amazing. So, okay, here I'm cheating, this is from maybe December, the beginning of December. So, okay, you can say it's not that cold, but this is a picture that I took today. So for July is not the best. So, and this half of the summer is like that, if we are lucky. So one of these cold winter nights, I was thinking, okay, what I'm doing here? So then I thought, okay, maybe cold temperature could be the solution or the answer to our question. Why? Because, of course, it's known that extreme cold can be detrimental for the organism, but it's also known that a moderate decrease in cold temperature can actually be beneficial. Why? Because cold temperature reduces the rate of chemical reactions and metabolism, and that results in slower molecular entropy, energy expenditure, and also a slower pace of healing. So actually, cold temperature is one of the most effective mechanisms to extend lifespan in many organisms, like worms, flies, fish, and even in mammals, in mice. So for instance, in C. elegans, the average, the normal or standard temperature in the lab is 20 degrees. So at this temperature, the worms live around 19 days of average. If we put the worms at 25 degrees, they live much shorter. But if we decrease the temperature to 15 or 10 degrees, then they can live up to 3 times longer compared to the control temperature. And what I would like to remark is that these worms look very happy. They are moving around like they were basically young. So we couldn't see any detrimental effect induced by cold temperature. So we think that, so this led to our hypothesis that maybe at low temperature, there's less pressure to prevent or repair damage to the different tissues, and also there's less need to spend energy. So that we think that facilitates the distribution of resources between tissues. So we thought that maybe under this condition, at cold temperature, there is no need to sacrifice the soma or the germline, and also that there's a rewiring or adaptation of genetic programs and communication between tissues that are involved in the regulation of aging and longevity. So to test this hypothesis, we went back to to the GLIP1 mutant, the one that don't have a germline. And as I said before, these worms at 20 degrees, they live much longer compared to the control strain. But we obtained very exciting results when we put the worms at 15 or 10 degrees. So under this condition, we found that the GLIP1 mutant worms live much shorter compared to the control strain. So that was very exciting because it seems that the germline is actually required for the co-induced longevity phenotype. So in worms, the germline, the adult germline, contains a mitotic region followed by different cells in different stages of meiosis that finally generate gametes for reproduction, like for instance sperm and oocytes. So what is important to say is that there is a continuous loss of germ cells during the aging process due to gametogenesis and apoptotic events. But to avoid this, the mitotic region contains a pool of adult germ cells with all the characteristics of stem cells. They can self-renew, they can proliferate, they can differentiate into gametes, and they also, so they basically replenish the pool of germ cells and oocytes. So the function is to rejuvenate the germline. So by BRDU staining, that only stains proliferative cells or dividing cells, we found that at 25 degrees, there's a rapid decline in the pool of germ cells. So basically, at day 5, it was already very difficult to find germ cells, and they were completely gone at day 7. And at 20 degrees, there's also a rapid decline in the pool of germ cells. At day 10, it was already hard to find germ cells, and they were basically gone again at day 15. But at 10 degrees and 15 degrees, they can maintain the pool of germ stem cells for extended periods of time. For instance, at 15 degrees until day 25, and at 10 degrees until day 37. So, as I said, the germ cells are critical to maintain the pool of oocytes, to generate new oocytes, so they are critical for reproduction. So we found that during the first 4 days of adulthood, when the worms are young, there were no massive differences in the number of progeny that the worms can generate at different temperatures. But when we studied this at day 10 or beyond, then we obtain dramatic differences. So for instance, at 25 degrees, after day 10, they don't lay any more eggs. At 20 degrees, they lay a few eggs for just a couple of days, but at 15 degrees and 10 degrees, they lay eggs for extended periods of time. And also the total number of eggs that they can lay at this cold temperature is much higher than the one that you can find at 20 degrees. So it seems that indeed cold temperature maintains the pool of germ cells. So the question now was how the organism regulate this maintenance of germ cells. So recently it has been discovered that TRP1, a colon-sensitive channel, is required for colon-induced longevity, and that this TRP1 channel is only expressed in specific neurons called IL-1 neurons. So as this has been published, so we just repeat it. So we found that indeed TRP1 mutations decrease colon-induced longevity, but overexpression of TRP1, specifically in IL-1 neurons, was sufficient to rescue this short life phenotype. But the most interesting for us is that these TRP1 mutants don't show any effects on germ cell maintenance at day 5 when the worms are still young, but TRP1 channel becomes critical to maintain the pool of germ cells at older ages, like at day 9. And we could rescue this low number of germ cells if we overexpress TRP1, specifically in this IL-1 neuron. So we can conclude that indeed the neurons or the soma can regulate the pool or the maintenance of germ cells. So with these results, we ask, okay, can the germ cells in turn or reciprocally regulate the aging of the entire organism? So for that, we treated the worms with FUDR. That is a compound that inhibits DNA synthesis. So basically, it inhibits or it particularly affects proliferating cells. So it's important to say that germ cells is the only pool of proliferating cells in adult worms. So we thought that FUDR treatment specifically would affect these germination cells. So indeed, by BRDU assay, we found that when we treat the worms with FUDR, we could completely eliminate the pool of germination cells above that 20 and 25 degrees. But the most exciting was that when we treated the worms with FUDR at cold temperature, we could completely block the cold-induced longevity. In contrast, that didn't affect lifespan at 20 degrees. So to further test or confirm these results, we decided to note down orthologs of 5A, the translational factor 5A, because worms have a particularity that they express two orthologs of these factors. One is IF1, that is only expressed in the germline, actually is necessary for germline maintenance, and IF2, that is only expressed in somatic tissues. So we let the worms develop into adults with a normal germline, and then we initiated the knockdown of these factors, and we found that indeed IF1 decreases the pool of germination cells, but IF2 doesn't have any effect on germination cells. And in this line, we found that IF2 doesn't shorten the lifespan of the long lifespan at cold temperature, but IF1 RNAi completely block this cold-induced longevity without affecting lifespan at 20 or 25 degrees. So taken together, the results indicate that the germline can communicate with the soma and change the soma to extend longevity. So now we wanted to define what are these changes. So for that, we perform proteomic analysis with worms at different conditions, cold temperature plus IF1 RNAi, and worms at higher temperature. So the idea was to identify those proteins that decrease upon IF1 RNAi or upon an increase in temperature. So after we identify these factors, we perform an RNAi screen to see if they are important for cold-induced longevity. And by using this approach, we identify a handful of interesting candidates that are required for cold-induced longevity, like CCTs, AMA1 or CBS1, but they don't affect lifespan at 20 or 25 degrees. So we thought that these factors could affect longevity by two different mechanisms. First, maybe they directly regulate germination cell proliferation, and that eventually affects the longevity of the organism. And the second, and most interesting for us, is that germination cells upregulate these factors in somatic tissues. And this is what extends lifespan. So to discriminate between the two options, we perform BRDU experiments, and actually we found that most of the candidates fall into the first group. They can decrease the proliferation of germination cells. And likewise, if we note down specifically these factors all in the germline, they also decrease cold-induced longevity. But the most interesting for us was CBS1, that is an ortholog of human cystathionin beta synthase, because we found that knockdown of CBS1 doesn't affect the pool of germination cells, and also when we note down this factor all in the germline, then that didn't affect lifespan. So that indicates that CBS1 is actually acting in the soma to extend longevity. So indeed, we found that if we note down CBS1 in the muscle, in the intestine, we could decrease the cold-induced longevity, and this seemed to be specific for both for muscle and intestine, because knockdown in neurons didn't have any effect. So then we generated a transcriptional reporter of the expression of CBS1, and we found that at both 10 degrees and 15 degrees, there's a higher levels or higher expression of CBS1 compared to 20 and 25 degrees. And this expression was mostly linked to the muscle, and particularly to the intestine. And when we treated the one with FUDR or IF1 knockdown, we could decrease these high levels or this high expression of CBS1 in the somatic tissues. So that indicates that, yes, the germ cells are regulating the expression of CBS1 in the intestine. So how CBS1 regulates longevity? So CBS1, as I said, it produces cystathionin, but one of the byproducts of this reaction is hydrogen sulfide. So hydrogen sulfide is a gas, similar in molecule, with important roles in longevity. First, it can prevent neurodegeneration. It's also required for longevity induced by dietary restriction in invertebrates and rodents. And also, the most interesting for us is that just by adding S2S to the plates, we can extend longevity. And as you can see, the longevity phenotype is quite significant. So then we measure production of S2S. And as you can see, at cold temperature, 10 and 15 degrees, the worms produce more S2S compared to warmer temperatures. And these high levels could be decreased when we knock down CBS1. And most importantly, we found that if we knock down IF1 or we treat the worms with FUDR, we could also decrease these high levels of S2S. So that indicates that the germ cells are indeed modulating the levels of S2S at low temperature. So then we try to mimic what happens at cold temperature, but now at 20 degrees, just by overexpressing CBS1. As you can see, overexpression of CBS1 is sufficient to increase the production of S2S. And we found also that a specific overexpression of CBS1 only in the intestine induced the longer lifespan extension compared to, for instance, muscle or humans. And that happens at both 20 degrees and even warmer temperature. So it seems that by overexpressing CBS1 in the intestine, we can mimic what is happening in a cold temperature. So finally, we asked how the germline communicates with the intestine to extend longevity. So in other words, what are the signals that they release to modulate the intestine? So for that, what we did is to isolate the germline and perform transcriptomic analysis under different conditions. And we found that both IF1 knockdown or an increase in temperature decrease the expression of PGS2. PGS2 is a prostaglandin E2 synthase that synthesizes prostaglandin E2 that is the most stable and efficient of all the prostaglandin hormones. So indeed, we found that PGS2 mutants live much shorter at 10 and 15 degrees at cold temperature, but they don't have a lifespan at 20 or 25 degrees. So it seems that PGS2 is required for cold-induced longevity. So then by Western blot, we confirmed that at cold temperature, the worms indeed express higher levels of PGS2. And by immunostaining, we found that indeed the germline is basically the tissue that expresses this enzyme. As you can see, they express higher levels compared to the intestine. And also that cold temperature, 15 degrees, induces the translocation of the PGS2 enzyme from the nucleus to the plasma membrane, where it's supposed to be active. And in this line, supporting this experiment, we measured the levels of prostaglandin E2 hormone, and we found that indeed at cold temperature, the worms produce higher levels of this hormone compared to 20 or 25 degrees. So PE2 hormone is normally is specifically released by a specific transporter called MRP6. And we found that when we knock down MRP6 at cold temperature, that was sufficient to decrease cold-induced longevity, but doesn't affect longevity at 20 degrees. And also, we found that if we knock down MRP6, specifically in neurons or in the intestine, there was no effect on cold-induced longevity, but if we knock down MRP6 only in the germline, we could completely block the cold-induced longevity. So that indicates that the germline is in charge of releasing the prostaglandin hormone to promote longevity. And to support this data, we performed rescue experiments. So first of all, we treated the worms with FUDR to decrease the number of germ cells and the expression of CBS1 in the soma. But then we found that just by adding exogenous amounts of prostaglandin E2 to the plate, we could rescue the expression of CBS1 and also the short lifespan induced by FUDR treatment. So finally, to conclude, we have seen that neurons can detect low temperature, and then they communicate with the germline to maintain or sustain the pool of germ stem cells for longer periods of time. And of course, that decreases reproductive aging and extends longevity. But at the same time, the germline releases prostaglandin E2 hormone that is uptaken by the intestine to increase the expression of CBS1 that produces more S2S, and that extends lifespan. So these results were very important because for the first time we showed that the germline can actually promote longevity depending on the conditions, and they do that by releasing prostaglandin that coordinates extended fecundity with extended longevity without sacrificing the soma of the germline. So now I would like to thank my lab, especially Hanju Lee, who performed the prostaglandin E2 experiment project, Brando, Seda, and Isa, who work on the stem cell part, human stem cells, and of course the rest of the lab with this convenient picture here in the winter in Cologne. So I hope that all of us live quite long. So with that, I would like to conclude my talk, and now I will be happy to take all your questions. Thank you. Lourdes?
Thank you. I was looking for the microphone to reopen it.
Sorry, it was long, but it's very difficult to give a seminar without seeing the faces.
No, that was very. Thank you very much for this very interesting seminar with very brilliant results. There are several questions in the chat, which I will now read to you. Let me see if I can. Oh, for me also, it's difficult.
No problem.
So the first question is by Esther Serrano. Could you do a genetic screen in C. elegans Huntington model to find other regulators? The readout could be lifespan or locomotion.
Yes, of course. I mean, we have the library that covers 90% of the genes, so we can do a knockdown of basically 90% of the genes, and of course, for lifespan, maybe it's not the ideal one because probably she knows how it works. You have to transfer the worms every day. So for each condition, it should be more than 100 worms. And the problem is that they live very long at cold temperature. So it's not the fastest experiment. So, but she said we could look at other parameters for sure. And I'm sure there are many other mechanisms involved in this longevity, co-induced longevity.
Mm-hmm.
I don't know if I answer properly there.
Before I move to another question in relation to this, I was also wondering how does this translate to what could be done, not specifically for aging, although this is a disease we will all go through it, but more for Huntington and so forth. Would it be better to check for drugs that are already known as proteostasis activators, or maybe activators of those genes and proteins that you have found to be involved in proteostasis decline and so aging, or Huntington disease aggregation of the toxic protein?
Exactly. So that's basically, now there are one postdoc working on that, more in the link with proteostasis, because we saw also changes in proteostasis, and that maybe the effect wasn't that strong in lifespan, but there was increase at cold temperature and this. We are trying to, I mean, for us, the most convenient is the filter trap assay, because then we can look aggregation very fast and this. And we are working, yes, we started with Huntington disease. We have models for other diseases like ALS, I mean, many diseases. So, yes, so the first will be always Huntington disease, but we are also interested in other diseases. So
well, there are many conformational diseases where proteostasis need to be activated to eliminate toxic aggregated proteins. So,
so that's actually one of the aims that I had in my last grant. So actually, so
we have another question from Antonio Gil. Does the germline relationship with longevity hold up when compared to other model organisms, or is it very particular for C. elegans?
The first part I didn't get it. Which protein?
Does the germline relationship with longevity hold up when compared to other model organisms, or is it particular for C. elegans?
Yes. So in our case, for our project, I mean, what I can say that is calling longevity. I mean, calling this longevity is actually even seems to be important even in mammals. It also works in mammals by different methods, of course, not regulating the germline. So more specifically, how the germline affects longevity. So that of course it was first discovered in C. elegans with this genetic intervention that is for C. elegans is a very convenient genetic system. There is also publications in Drosophila that say that also a decrease in the germline can also extend lifespan. But in Drosophila is not that clear, I must say. And there's a recent paper now, PNS, that where they found this in zebrafish, that removing the germline is also modulates the lifespan. What is known in mammals and this is even in nature is that regimes like paradigms that reduce fecundity, I mean, without removing the germline, but just reducing fecundity, can extend lifespan, even in nature. So for that, I don't know. In my case, I don't know. We have these plants, that I don't know how they are called, that they have this mini. Later, like, they reproduce with mini plants, and it was very obvious. The plant was kind of dying. We removed this, and then became much healthier. So I think it could be a conserved mechanism in many species.
Okay, so let me see. Another question by Esther. How do IL-1 neurons communicate to germ stem cells?
That we don't know. That's a good point. I mean that was it was I mean the TRP1 channel was published in 2015 by Sean Shu. They are in US. It's a very good lab. After that, they had several publications in this topic, and in the last one was published in Genes and Development or something like that. There was a very comprehensive study, and they found that these IL-1 neurons release. I cannot say it does for the germline because they were studying neurons downstream directly and other tissues, but not the germline. So I'm not sure which factor exactly, but it was a neurotransmitter, but I don't remember. But I don't remember exactly which one it is. But of course, I don't know if this is the way how they communicate with the germline. I think there might be also some intermediate steps or tissues that could transmit the signal.
And in relation to this, a question by Jose Antonio Esteban, do you know how these IL-1 neurons control germ stem cell line viability?
Sorry, how do
the IL-1 neurons control GS cell viability?
Yeah. So as I said, It's very mysterious system. I mean, very interesting, but to be honest, we have no clue. So I think the first experiment that I would do is to follow this previous paper I told you, and see if these signals are also important for that. And for that, we already have the mutants, and this lab was very collaborative on this. So we have all these tools. So they even know which kind of vesicles they use for this communication. So for that, it's very convenient. So we can start with that. But I mean, the honest answer is that we have no clue at the moment.
Okay, another question by biodiversion. Thanks for your talk, David. Very interesting. I was wondering whether the overall turnover of proteins is altered during aging and or during cell differentiation.
Yeah, this is also a very interesting question because what is also, it was recently published that, well, not that recent anymore, in 2015, that actually stem cells also need high levels of global protein synthesis rates or relational rates. So they need to synthesize, to be very active synthesizing proteins, and if there's some small decrease on that, they start to differentiate or die. So it's like having higher proteasome activity. So it's so with, of course, both processes could be related. So one, maybe proteasome activity is also important in stem cells to be able to degrade the high amounts of proteins that the stem cells synthesize by default. And so more specifically for the questions, so what is known is that stem cells, basically, I mean, the chromatin are very open, they have high translational rates, and basically they are ready to express everything. And actually they express, besides the proteins that they need in high amounts, like pluripotency factors, they express also little amounts of almost every protein. So here the proteasome could play an important role, but more specifically for the turnover, I must say that that was something that we wanted to do when I was, after we published my papers in San Diego, because we have also a good proteomic lab there, and we wanted to see if these changes in the turnover with differentiation, but we don't know, and we didn't do the experiment. So I always try to see someone is working on that and see someone continue that, but I haven't seen any data in this regard yet.
One question from Esther: have you tried SID1 mutants?
Yes, but we tried the SID1 mutants only for, I mean, the ones where we recover SID1 over levels in specifically in neurons, to basically have a knockdown, to basically have only efficient knockdown in neurons. But I don't know why she asked that, but maybe I can help me to answer, or if I already answered, I don't know. But we use, we use.
And one last question by Paola Bobolenta. In humans and other organisms, body temperature is internally controlled. In this view, how the finding of the relevance of temperature in C. elegans could apply to other organisms?
Yeah. So, so I mean, actually, that was one of the reasons I decided to go to San Diego, but not. I mean, that's a funny story. So I applied to 2 labs. One was Andy Dillon, the one where I finished doing my postdoc, and the other was the lab of Bruno Conti. And actually, I interviewed there first. and I really like it. And I went to that lab because that was already in 2009, and I read a paper from him in 2005, where they basically, of course, mammals can regulate the temperature, and so you can put them at 0 degrees, so the internal temperature won't change that much, or won't change. But what they did in these experiments is to trick the thermostat in the brain of the mice. So basically they thought that they have, by tricking the thermostat, the animal thought that it had 30, just half degree less, and that extended up to 20% the lifespan. So of course, to translate that to mammals is we are not ectotherms. So we have strong mechanisms to regulate our internal temperature. So in this case, they do a mutation from pump neurons that regulate the temperature, that sense and regulate the temperature, and with that they could treat the organism to live longer. So what is known is that, and now this is more correlative studies, there are some, as I said, I mean, we should take that with pinzas, no, like we say in Spanish, that they show that cold temperature could be actually related with lifespan, or with longer lifespan. And another factor, that there are many papers that say that, but most of them correlative, that say that a decrease in menopause, or women that have menopause in later stages, they can live longer. So basically maintaining the reproductive system for longer periods is associated to a longer lifespan. But of course, this is all correlative.
Okay. I think there are no more questions. I would like to thank David and all the people who were connected. Up to 100 people were connected via YouTube. David, now there are 80, 90. So thank you all for this webinar. We will put it in our YouTube channel for you to review it. And now we will.
I don't think that we'll go through this punishment.
No, but maybe to see for specific results. And now I will finish broadcasting by YouTube. Thank you, all of you. And David, please stay for some minutes.